Jai Sri Ram, Jai Sri Ram, Jai Jai Sri Ram. Hello everyone, my name is Gyanendra Kumar. I am biology educator in India. In this video, we will discuss difference between prokaryotic genome and eukaryotic genome. Fine. We all have a basic idea about the prokaryotic and eukaryotic genome. We all know that genome is total sum of DNA of cell. Yeah, total sum of DNA of cell, isn't it? That is only genome. So we all know that our prokaryotes have circular DNA in their cytoplasm. Yeah, they have circular DNA in their cytoplasm. This DNA is obviously double stranded DNA. But eukaryote, you will see in the eukaryote have nucleus, like this is the nucleus of the eukaryote, and inside this nucleus chromatin network present. And when this chromatin network decondense, then we observe that in this chromatin network lot of chromosomes are present, and this in this chromosome nucleosomes are present, and in this nucleosomes DNA are present in highly condensed way. Yes, right. So our prokaryote have also DNA as a genetic material, eukaryote have also genetic material, both are genome actually. But we, as we discussed, that prokaryotic DNA, prokaryotic chromosome, their genetic material is actually circular DNA. Yeah, they have circular chromosome, they have circular DNA, but eukaryote have linear DNA. Yeah, eukaryotic DNA is actually linear, students. Okay, one more thing we have to understand, eukaryotic cells have mitochondria. So that is exception actually. Eukaryotes have mitochondria and eukaryotes have chloroplast. Yes, they have chloroplast. Mitochondria have also circular DNA and chloroplast have also circular DNA. But major DNA which is present in nucleus, that is actually linear DNA that is present in their chromosome. Fine. So they have which kind of chromosome? They have linear chromosome, linear DNA present. Right. And then we observe that our prokaryotic chromosome, monopoloid. Okay, only one set of the each gene present, right? They are diploid, they have actually two set of one gene. For each chromosome they have two copies, right? Okay, so one gene have how many allele? Two allele, at least in generally, I mean, see, in generally they are diploid. That means for one gene they have two alleles, right? And total two set of chromosomes are present, and in that chromosome DNA present, right? Okay, fine. In prokaryotic cell, they can have plasmid frequency. What is plasmid? Plasmid is one kind of extra chromosomal DNA which is not essential for survival of that cell, right? So often it will be present into the prokaryote, not in every prokaryotic cell, but often this extra chromosomal DNA, this plasmid, is present in prokaryotic cell. But plasmid, most of the time it's absent into the eukaryotic cell. There are some exception, obviously, like 2 mu circle. This 2 mu circle is present in fungi. Circle is present in fungi. There are some exception, but eukaryotic cells have not their. I mean, they don't have. I mean, plasmid. Okay, next you see the prokaryotic DNA. If you see this prokaryotic DNA, the prokaryotic, because prokaryotic cell have not plasma membrane, and the process of formation of the DNA is called replication. And from, like, we have one DNA, yeah, this is one DNA we have. Okay, and when they have to form another copy of the cell, any organism I'm saying, they have to form another copy of their DNA. This process is called replication. So they will replicate, and after replication from this DNA they will form RNA, and from RNA finally they form protein. The process of formation of RNA is called transcription. The process of formation of protein is called translation. Is called translation. Fine. So our prokaryotic DNA are have not nuclear membrane. So that either it is transcription or it is translation or DNA replication. This everything will happen in cytoplasm, because they have only one chamber, that is cytoplasm. Okay, but if you see eukaryotic cell, in eukaryotic cell the DNA replication occurs in the nucleus, transcription occurs in nucleus, but translation occurs in cytoplasm. Yes. Then the chromosome, is present into the prokaryote, is present in the condensed form in the nucleoid. Okay, and they don't have histone protein, and their genome is smaller in size, and they don't have interon. What is interon, students? In a DNA sequence, DNA sequence is huge chain of the nitrogenous bases. In that sequence there are some region, they will not form messenger RNA, and that regions are called interon. Non-coding part of the DNA is called interon. So interon is almost absent. Very less non-coding region present into the prokaryote, and this prokaryote, then we discussed, they have small gene and they don't have a stone protein, right? But if you see the eukaryote, they have membrane bound nucleus and they have histone protein. Okay, and then their genome is larger in size. Their genome is comparatively larger in size, and they have lot of interon, means non-coding, lot of non-coding region present, right? Then that non-coding region is what? They are repeated DNA sequence. Try to understand it. Like I have one this eukaryotic DNA. So the portion of the eukaryotic DNA which will form protein, I mean which have to form RNA, that will be called exon. Yeah, and the remaining portion is called interon. So in eukaryotic cells, lot of interon is present because DNA, is comparatively, it's huge, it's long in size due to presence of non-coding, lot of non-coding DNA. Okay, let's compare more actually. If you'll see the ratio of the genetic ratio and the, I mean the, you will see the ratio of the coding and non-coding region. So 90% of the protein coding genes are present here in prokaryotes, right? Only very little non-coding DNA is present, but in eukaryote you'll see only 5 to 10% are protein coding regions. Remaining all, is non-coding actually. They have lot of pseudogenes and different type of repeated sequence are present. They are important actually. They protect the coding sequences, right? Now if you'll see the gene structure, see here the gene structure is simple, continuous, and they have very little interon. Here they are modular. Okay, and gene contain interons, and lot of untranscribed regions are present. Untranscribed region, we cannot participate in transcription. And then you see transcriptome. What is transcriptome? If we have a genome which is sum of the all DNA, I'm saying, so whatever the total sum of RNA they form, that is called transcriptome. The total sum of RNA formed by a genome is called transcriptome. So here transcriptome is simple. They are mostly messenger RNA, but here in another side in eukaryote you'll see they have great abundance of non-coding transcripts. Okay, and that non-coding transcripts, they will maybe they will participate in regulation of the whole process. Okay, then if mobile element, you see they have little number of mobile elements, they have lot of mobile elements are present. Okay, then if you will see you will see the DNA changes here. So they have lot of mutation. They will go through, I mean our prokaryote will go through lot of mutation based changes, right? Okay, and you will find horizontal gene transfer also there. So by the horizontal gene transfer, they will acquire new genome. But here actually, here if you'll see basic evolutionary process, they have high content of the repeated DNA sequence, which will facilitate recombination, translocation, shuffling. Yeah, see, this recombination, translocation, and shuffling and all, it's present into the our eukaryote mostly. Recombination, you see, like in the our meiosis, recombination occurs, right? And sometimes there will be translocation of part of genome across the chromosome also, so that all things can be seen. Okay. And then if you'll see mid evolutionary tendency, genome will show compactness here, and they will show adaptivity here, but here genome will show expansion and complexity, I mean in eukaryotes. So in this video we discussed that our prokaryotic cells have less non-coding area, eukaryotic cell have more non-coding area. The prokaryotic DNA is smaller in size, eukaryotic DNA is longer in size. Prokaryotic DNA have less interon, eukaryotic DNA have lot of interon. Then mobile elements are present very less in the prokaryotic cell, more lot of mobile elements are present in eukaryotic cells. Then we saw that our prokaryotic cell in DNA changes, they will reliable mainly on mutation and horizontal gene transfer. But in eukaryotic cell, the DNA will change by the recombination, translocation, and shuffling, even duplication, and rearrangement of the different rearrangement of DNA in the different way. And then we saw that there will be in the main evolutionary tendency in the prokaryote is the genome adaptivity or genome compactness. But here is the genome expansion and complexity. In this video, till here only, please subscribe this channel for coming so many interesting videos. See you next video. Namaskar.